Okay, guys, so on your own on Tuesday or Wednesday, you looked at big ideas 7 and 8, which are both looking at the innate immune response when some kind of bacteria, virus, pathogen enters the body. And the innate immune response is primarily, or is only, first lines of defense and second lines of defense. And so those happen no matter what enters our body. Anytime something enters our body, those are the first to kind of fight it off. Now, if this pathogen gets further into our body, deeper into our body, and is spreading more quickly, it's beat those first line of defenses and second line of defenses, then this third line of defense is going to pop on in. So let's title these notes. Unit 5, Lecture 4, Big Idea 10. I know we didn't do a 9, but we'll talk about why later. Big Idea 10. And that is to explain both cellular and humoral adaptive immune responses. So the first thing I want you to do is fold your paper in half, hot dog style. And both humoral and cellular adaptive immune responses are initiated once this pathogen has entered our body and moved past our first and second line of defenses. And so we're going to look at both of these. We'll put antibody-mediated immunity, which is another name for humoral responses, on the left side. So antibody-mediated immunity. And then we'll just define it below. And it's another name for humoral immunity. And this is everything that happens here is going to be what's going on when this pathogen is in the extracellular fluid. Right? So with like SARS-CoV-2, we saw how it binds to a cell and then it kind of moves on into the cell and replicates inside the cell. This is for when the pathogen is not inside a cell yet. It hasn't latched on. If it can latch on, it will eventually, but if it hasn't latched on yet, we can fight it off in the extracellular fluid before it's actually inside a cell, extracellular around the cell fluid. Sometimes that's blood, sometimes it's just between the cells in the tissue, so in those types of times. And then on the right side, we'll label cell-mediated immunity and define that one as cellular. And it's when the pathogen is in our cells. So we're going to start off, I'm going to use colors. If you don't have colors or you're in school and don't have them, you don't have to use them. It's totally optional. And the first color I grabbed was green, and my green marker is dead, so let me grab a different one. But we're first going to start by drawing a pathogen in the middle, and so we're going to make this kind of like a cartoon. And so there's different types of viruses. Here's one type. It's called the bacteriophage. We've also seen coronavirus, another type, which is a very different shape from what we're drawing right here. And we can label this our pathogen. Again, pathogens are anything like bacteria, viruses, fungi, any foreign agent. Okay, so the first thing that's going to happen when this bacteriophage has made it past our other stages of defense is eventually we will have a macrophage come over and phagocytize it. So we're going to draw a macrophage. I'm going to use yellow because these always remind me of Pac-Man. And this macrophage is going to engulf our pathogen. So I will draw this pathogen inside its engulfing mechanisms. Looks like its mouth, looks like it's eating it, even though it's really not what it's doing. And I'll draw a couple of pathogens inside as well. Some of them in pieces, others as a whole. And it phagocytizes these pathogens, and then what it wants to do is present it. And so I know I said that macrophages don't primarily present, but they do. So I was a little off on that. But anyways, here it's going to go and present this on the outside of its cell. So it's going to present a piece of this pathogen. Okay. And it's going to basically go around telling all the other blood cells, it's floating through the blood, and telling all the other blood cells, come look for this. Attack it, look for it. This is what I have found. It's kind of like if you've ever seen or heard about like those tribal cultures where they kill like those that they're fighting against, and they use their skull or their bones and make necklaces to showcase what they've killed. That's what this is doing. It's like, hey, look at me. This is what I got. Go get it as well. Okay, and so we'll just define this underneath and say macrophage, phagocytize the pathogen, and they present it. Okay, so this happens no matter when a pathogen enters our body. This happens in both antibody-mediated immunity and cell-mediated immunity. So from here, what this macrophage is going to do is it's going to go around and it's going to bump into tons of helper T cells. Okay, and so I'm going to use red for my helper T cells. And what helper T cells are going to do they're going to interact with antigens. Antigens are part of a pathogen, so like a little piece of it, like is shown here. So they're going to interact with antigens on macrophages and do a couple of things. They're going to produce cytokines and activate T and B cells. Okay. So from here is where we're going to branch off. Okay. If we are dealing with a pathogen that has infected our cells, we're going to go towards the cell-mediated side. And I'm going to draw some just little red dots here, which are showing that the cytokines are heading this way. They're attracting other cells and calling them over so that they can then activate them. There will be T cells floating around in our blood. There will be B cells floating around in our blood, but they do not do anything until they are activated by a helper T cell. So they must first come over to this helper T cell, get activated, and then they can go do their job. So these cytokines that we just drew in red, I'm going to label, they attract other cells to the area to be activated. Okay. They're kind of like that whole analogy to a guard dog, where the guard dog's barking and it draws attention to the scene, and others come. It might be like, oh, there's an intruder, and then others are going to come over, or the person whose house it is going to look out their window and see what's going on outside. What's the dog barking at? You can think of it as a car alarm or a house alarm. And if the fire alarm goes off or an alarm goes off or a car alarm goes off, people look. They are drawn towards it. They're seeing what's going on over there. That's what these cytokines do. So what they're going to attract and activate are T cells. Sometimes they're killer T cells, other times they're cytotoxic T cells. So we'll draw a little arrow down and we will draw our cytotoxic T cell. Now these are also T cells, so I'm going to use red again, but I'm going to draw them a little bit differently. I'm going to draw them with some spikes because these are going to go up to our body's infected cells and poke them. So here's their poking spikes, and then inject them with something called perforin. So kind of like little assassins. I'm going to give it some like shades. I don't know. There we go. Okay. So this is our cytotoxic T cell. Cytotoxic T cell. Okay. And what it's going to do is bind to infected body cells and inject with perforin. Okay. Inject with perforin. And that's a toxin that's going to cause the cell to lyse. It's going to kill our own body cell and all the other pathogens inside of it. Why does it do this? To prevent spread of the pathogen. Now, when you play your game tomorrow, you're really going to see how important this is. Why, if we don't have these guys at work, this pathogen is going to spread really, really, really fast. There's actually a virus called HIV, which I'm sure you've heard of. And what it does is it actually hijacks the cytotoxic T cells. And so it doesn't allow them to do their job, which is really, really difficult for our body because when our T cells aren't active, pathogens spread really quickly. Viruses spread quickly. Bacteria spread quickly. So even a common cold could kill someone if their T cells aren't active, meaning HIV doesn't usually kill people. It's anything else, like the common cold that kills people because their T cells are inactivated and can't fight it off like normal. So these are huge in protecting us. So this is a pathway we take if the pathogen, again, is inside of our cells, it's infected our cells and making copies of itself in there. Now, if we go back to here where we had our helper T cells that are going to activate T and B cells, that was the T cell path. Now we're going to look at the B cell path. If this pathogen or bacteria or virus is in the extracellular fluid, it's not in our cells yet, it's just floating around. And so we're going to head to the left. And I'm going to add some more dots here to show that cytokines are going to call over these guys as well. Both of these would be happening often simultaneously because the pathogen's probably in the extracellular fluid and in our cells both at the same time. I'll just label these, we don't have to define them again, as cytokines. And over here I'm going to use blue. Because now since we're not working with T cells, we're working with our other lymphocyte called B cells. It's going to call B cells over. It's going to attach to them. It's going to activate them. And once they're activated, they're actually going to differentiate. Just like those monocytes, when they leave the blood, differentiate into macrophages and dendritic cells, we're going to have our B cells copy. And so they're going to make copies of themselves. And then they're going to differentiate into different types of B cells. So B cells duplicate. And they are going to make two different types of B cells. Some of these are going to be plasma B cells. Which are going to produce antibodies. And antibodies are little proteins that look something like this Y shape. And we have lots of different types of antibodies. Antibodies are also called immunoglobulins, so you'll also often see them as written as Ig, immunoglobulin, immunoglobulin M, IgM, IgG, IgD, IgE. And each of these has a slightly different function in terms of what they're going to interact with. Whether it's a bacteria or a virus or a transplant cell. Okay, so there's a couple of antibodies. And antibodies have a couple of roles themselves. Antibodies are going to, number one, bind to the pathogen in the extracellular fluid and prevent it from moving. Now this is huge because if our pathogen can't move, it can't get into our cells and it can't copy itself, and that saves us. So it basically indirectly kills the pathogen by just holding it in place. But it itself can't do anything other than prevent it from moving. What's going to have to happen next is a macrophage is going to have to come along and then engulf and break down this pathogen after it's held and stuck there. But at least it keeps it from moving, which is huge. And after it does that, it's going to attract other cells to come break that pathogen down to ingest the contraption it just made. I like to think of antibodies as a couple of things, like a boot on a car. If you've ever seen a car boot, it can't move. It's stuck there, can't go anywhere. A spider web, right? A spider web traps insects, but it can't do anything else, and the spider has to come in and take care of the insect and eat it. I also have had some students compare it to like a third wheel on a date. It's kind of like holding everything, holding you down. I don't know. Anyhow, that's antibodies. And then the other type of B cell that will form when these B cells differentiate are called memory B cells. Oops, I forgot to draw the plasma B cells. That's okay, we'll just draw it here. And here's our memory B cells. You just add like a line there, 2 lines here to show they're slightly different. Now memory B cells are going to remember our little pathogen for the future. Okay. So these have a pretty long lifespan, these memory B cells. So they'll remember them for a decent amount of time. So memory B cell remembers the pathogen. Okay. So I want to show you a little picture from your textbook. And this is, oops, here we go. A little graph that shows the antibody concentration after the first time it's been introduced and the second time it's been introduced. So if we look after the first exposure, so the first time a certain, like say SARS-CoV-2 enters our body, you can see that it takes about 10, maybe like 5 to 10 days for a significant amount of antibodies to be in the blood and helping out. Versus on the second occurrence, you can see like right away it shoots on up. We see lots more antibodies, and much quicker, maybe 1 to 2 days after exposure, we have those antibodies at work in the cell. Again, when you see the game tomorrow, you'll see why it's so important to have those antibodies fast, because how quickly they can stop the spread of infection. So it's much quicker the second time around. This is another thing to be mindful of when you think of those COVID tests. There's 3 types of COVID tests. There's the PCR test, which tests for the viral RNA, meaning it's testing to see if the virus is actively in you at the moment because it's looking for the virus's RNA. The other is the antigen test. It's looking for that spike protein on the virus. So again, looking to see if the virus is in you, but just using a different format. A little bit less accurate than the PCR tests. Those are both the nose swabs, the mouth swabs. And then there's the antibody test. And this one's where they're looking at your blood to see if you have antibodies, those little Y-shaped proteins that your cells made in response to SARS-CoV-2 floating around. Now, if you were to get an antibody test a couple days after exposure, there's a good chance the test is going to miss the fact that you had COVID. You really want to wait till 5 to 10 days after exposure if you're going to get an antibody test to see if you had it. Otherwise, the antibody test will not tell you if you have it currently necessarily. It could, but it also could give an inaccurate false negative. So really, with antibodies, it takes a couple of days for those to be produced, which again, is why the first time you get something, you get really, really sick because your body takes a long time to get those antibodies. It's slower to fight it off. We lose more of our own body cells. Your immune system's working harder to get through all those steps we just looked at just to get to those antibodies. Second time around, it releases those antibodies so quickly it stops that infection much faster and you don't get sick. Now, the only other thing I wanted to briefly talk about is vaccines. So with vaccines, I know a lot of you already know this, but what they're doing is often injecting you with a weakened or dead virus. And that causes all of these steps to occur. Really the goal is to get this memory B cell made when they're injecting you with that. But the weakened or dead virus makes it so that you're getting You're getting the virus inside of you. You're getting all of these things made in response for the future. But you're not going to get as sick because the virus can't replicate, it can't duplicate, it can't spread around your body. It's a certain amount that's being injected into you. With coronavirus, it's actually a little bit different. It's one of the first times they've ever done this where they actually are not injecting you with either of those. They're injecting you with SARS-CoV-2's RNA. And it's specifically the RNA that codes for the spike protein. So they're injecting your body with RNA. Your body's then going to translate that RNA into spike proteins, and it's making the spike proteins in your body, which are then going to be presented by those macrophages and cause all of this memory and antibodies in response to it. So that was the only other thing I think I wanted to talk about. I guess another topic too would be with these memory B cells. There's a whole parenting debate where some parents are all about making sure that their kids are washing their hands and super clean and they're protecting them from viruses and getting sick. And that's one way of parenting. Or the others is parents are saying, you know what? Let my kid eat dirt. Let them lick doorknobs. Whatever they want to do, they're going to do. Because if they get sick now, their immune system's stronger, which is true. Your immune system's weaken and weaken and weaken as you get older. And so they're like, might as well let them get sick now when they can fight it off well. And then they'll have these antibodies for the future. And when they get older and they can't fight these off as well, they'll already be prepared. So, the safety way, they're hoping that their kids never get sick. But the preventative way, they're saying, well, most likely my kid's going to get sick in the future. Let's let them get sick now when they're stronger. All right, that's it.